This is passage 8. This is question 41. It asks, which outcome most likely results from exposure of these mutant mice to TNF alpha? So let's look at what OGG1 is in the passage and its relevance or its relation to TNF alpha. So if we look here, it says OGG1 is a DNA glycosylase that recognizes 8-oxo-G. It's involved in two processes, and the one we're interested in is this process right here. And then we should also keep in mind that it is also important for expressing basically this family of genes right here, and that's going to be important for one of the answer choices, and I'll get to why. But basically, it's basically important for repairing this mutation in the DNA. And how does this mutation form? It comes from here. Look at the first sentence right here. When you expose these cells to TNF alpha, it can create reactive oxygen species that create this mutation. So what do we have? We have TNF alpha causes this 8-oxo-G mutation. And how do we fight this mutation? We have this gene, OGG1, that recognizes this mutation and attacks it. So it wants to stop this mutation. It basically fixes it. And then a side note, what does it also do? It expresses these pro-inflammatory genes known as CXCL. So let's take a look at the answer choices here with that in mind. So when we have a mouse that is essentially none of this gene right here, this OGG1 gene, right? Because both chromosomes are lacking this gene. When we have these kind of mice and we expose it to TNF alpha, what do we expect? Let's take a look at A. It says an increase in cancer incidence. Well, let's go back up here. Remember, TNF alpha leads to this mutation, and what does this mutation do? It creates these GT base pairs into CA base pairs. So it's creating these mutations within the DNA. And what does repeated mutation do to the DNA? It can cause cancer, right? So we have TNF alpha causes cancer through this 8-oxo-G mutation. Now remember, we had OGG1 gene. We can use that to fight off this mutation, but what's wrong with our mice? They lack this gene. So what does that mean for us? That means we don't have anything to fight against this 8-oxo-G mutation, which means what? This TNF alpha could have a higher likelihood of causing cancer in our mice. So A looks pretty promising, but as per usual, we do want to look at all of the other answer choices here. B says an increase in CXCL2 expression. Now, if we increase TNF alpha, right, we might expect OGG1 to be expressed and create this increase in CXCL. But remember, we don't have OGG1, right? So this gene is essentially gone. And if that gene is gone, then we're not going to have anything to increase that CXCL2 expression, or at least it won't be increased from OGG1. So not that. C says a decrease in cellular ROS levels. Well, let's look over here. When we have TNF alpha, we are going to increase this reactive oxygen species, and OGG1 doesn't really do anything to stop that. And on top of that, we don't really have OGG1 to anyways in the first place. So if we increase TNF alpha, remember it says it produces or causes the cell to produce reactive oxygen species. So if we express these cells or these mice to TNF alpha, we're actually going to have an increase in cellular ROS levels. And finally, D says a decrease in KD of this compound or this molecule here, compound for its target. And remember, the target is the OGG1. Right? It looks like the 8-oxo-G structure, and it binds to the active site of OGG1. Well, if we express TNF alpha, the binding ability of this compound is not related to whether or not we have TNF alpha presence, right? This is just a compound that binds to OGG1. And on top of that, again, this OGG1, this DNA glycosylase, doesn't even exist. This mouse doesn't have the genes to encode for OGG1, right? It's a mutant. So we're not going to be really changing any of the properties of this non-existent DNA glycosylase in the mouse, since it doesn't even exist in the first place. We're not going to change the KD of this compound for its target, which is OGG1. So what are we going to do? We're not going to be doing that, which makes the most likely answer choice here A. All right, this is the second question of this passage. It asks, TH5487 acts as which type of inhibitor? So hopefully this is a little straightforward. This is essentially like a pseudo-discrete, right? You just have to know what these different types of inhibitors are. So let's look in the passage really quickly. TH5487, what does it do? It binds to the active site of OGG1. So out of these inhibitors, which one of them binds to the active site? That would be the competitive inhibitor, right? Competitive inhibitors bind to the active site of enzymes. So let's say this is my enzyme here, and this is the active site, this little pocket. What is this inhibitor gonna do? It's just gonna sit in the active site, and that's how it inhibits this enzyme. Now for review, let's just go over what mixed, non-competitive, and uncompetitive inhibitors are. Mixed inhibitors, okay, so these are inhibitors that can bind both to the enzyme itself or the enzyme plus its substrate. So what does that mean? Let's say I'm going to draw down here. Let's say I have an enzyme, okay, and this enzyme, let's say this is just the enzyme without anything, and then this is the enzyme with its substrate. So let's say this is a green substrate, okay? So what can this inhibitor do? It can bind to the enzyme with or without its substrate, okay? This is my inhibitor. Right? It can bind right there. It can bind to either at different affinities. Now, what if it can bind to both of these at equal affinities, right? This inhibitor doesn't really care if that substrate is there whatsoever. It can bind to both with equal affinity, then you will get a non-competitive inhibitor. So that's the difference right there, okay? Mixed inhibitors, that's just an umbrella term for any kind of inhibitor that can bind to both the enzyme and the enzyme plus its substrate, or the enzyme when it's bound to its substrate. Whereas non-competitive is a little bit more specific. That's a specific type of inhibitor that doesn't prefer either. It can equally bind the inhibitor, the inhibitor can equally bind the enzyme by itself without its substrate, and it can also bind the enzyme with its substrate at equal affinity. So that's what these two mean. But again, these cannot be the answer choices since that's not the definition of TH5487 that we're given. And then what does uncompetitive mean? Uncompetitive means that it can only bind the enzyme when the enzyme is bound to its substrate. So the inhibitor can only be in this formation where it binds the enzyme substrate complex. It can't bind it without its substrate. So again, the inhibitor can bind the enzyme only when the enzyme has the substrate bound as well. Again, that's not the definition we're given, so we know that A has to be the correct answer here. Okay, so this is the next question. It says, based on the data shown in figure 2, what are the effects of OGG1 and TH5487 on this gene expression CXCL1? So let's look at the options here. And well, first let's look at the figure. This is figure 2 right here. That's what we're working with for this question. Now, the first statement says OGG1 reduces the TNF-alpha-induced CXCL1 expression. Statement 2 says the opposite. OGG1 stimulates the TNF-alpha-induced CXCL1 expression. So first thing we can look at is here. We notice that if we just stare at this row, when we add TNF-alpha, we get increase of CXCL1 expression across the board, right? Regardless of whether or not they're wild type or they're deficient in OGG1. Now this question asks, okay, does OGG1 increase or decrease the TNF-alpha induced CXCL1 expression? So OGG1, remember the wild type, right? These wild type mice have the OGG1, whereas this mutant right here specifically doesn't have any OGG1. So we're comparing the black and the gray bands right here side by side. So let's look at this column right here, okay, these two columns. We notice that when we add TNF-alpha to these two columns, we see a far greater increase in the wild type, right? A far greater increase in the CXCL1 expression in the wild type mice compared to the mice that don't have OGG1. So what does that tell us? It looks like OGG1 is increasing the amount of CXCL1 expression when you add TNF-alpha. When you don't have OGG1, you don't get as great of an increase of CXCL1 when you add TNF-alpha into the mice. So what does that tell us? That OGG1 stimulates the TNF-alpha-induced CXCL1 expression. So 2 is correct, all right? And immediately we know that we can eliminate A and B. That's just a little test-taking strategy. Once you know answer choice is wrong, always take out all of the answer choices that have that specific option listed. So we know it's either C and D, so let's look at statements 3 and 4. 3 says TH5487 reduces the OGG1 mediated CXCL1 expression, and 4 says the opposite, that it stimulates, right, this CXCL1 expression. Same thing. Let's take a look at TH5487. So if we'll notice here, look at these two columns. This is really not interesting, right? Well, it is interesting, but it's not really going to help us because even if you add TH5487, really there is no difference in terms of the CXCL1 expression. So what do you want to look at then? We want to look at these two columns right here, okay? So it says OGG1 mediated CXCL1 expression. Now what are we comparing that? We're comparing these two black lines, this one right here and this one right here. Why? Because these are the wild type mice that have that OGG1 gene. Remember, these gray bands, they don't have the OGG1 gene. So if we're looking for OGG1 mediated CXCL1 expression, we're going to want to compare these black bands because that's the ones that have OGG1 in the mice. Now let's take a look. These conditions are the same, right? We're always going to compare everything else. When we compare to data points, we have to make sure all of the other variables that we're not really interested in are the same. So we hear both of these are TNF alpha plus, which means both of these mice are exposed to TNF alpha. And what we're trying to see is what happens when we add, right, the TH5487. What happens if we don't have versus when we do have TH5487? Let's take a look at these black lines. When we have TH5487, what do we get? We get a decrease in CXCL1 expression in the wild type mice when we have TNF alpha. So again, what happens? We add TH5487, we see a sharp decrease in the amount of CXCL1 expression in the wild type mice, which means what? TH5487 reduces, okay? It reduces the OGG1 mediated CXCL1 expression, which means D is incorrect. This is the last question of the passage. It says, researchers measured the NF-kappa B occupancy of the CXCL2 promoter following exposure to TNF alpha both in the presence and absence of TH5487. Based on the data presented in the passage, which figure depicts the expected results? Now let's take a look at the relationship between NF-kappa B and CXCL2. Right, let's take a look here. Remember, this transcription initiation of these pro-inflammatory genes, such as the CXCL family members, it includes the binding of NF-kappa B to the promoters, okay? So what does that mean for me, or what does that mean for us, is NF-kappa B increases, it leads to CXCL expression increasing. So what does that mean for us here? Well, actually, let's take a look at figure 2 first. First, let's look at what TNF alpha does. Remember, when we have TNF alpha, okay, when we compare the negatives to the positives, when we add TNF alpha, what happens? We see an increase in CXCL1 expression, okay? If we have an increase in CXCL1 expression, what this means is we have that NF-kappa B binding to the promoters, right? Remember, the increase of CXCL expression, or the expression of the CXCL family members, involves the binding of NF-kappa B to promoters. So if we see that we have an increase in CXCL, right, as we can tell from figure 2, when we add TNF alpha, it means we know that NF-kappa B is binding onto those promoters. So that's what it at least suggests to us, okay? So if we look at this right here, we know that TNF alpha, when we add it in, it increased to NF-KB occupancy, okay? Now, what about TH5487? Remember, when we compare these two data sets, okay, one without and one with TH5487, let's look at these bands specifically. What happens, right? When we have OGG1, what happens when we have TH5487? We see a decrease in CXCL1 expression. What does that mean for us? If we have a decrease in the CXCL1 expression, right, a decrease in CXCL1 expression, that means for us that NF-kappa B, that's not binding onto those promoters as much. So what does that mean? TH5487, okay, that leads to a decrease, okay, in NF-KB occupancy. Now, we see that even with TH5487, it doesn't completely make this CXCL1 expression 0, right? We compare it to when we don't have TNF alpha. We still see an increase regardless, even if we have TH5487, but that increase is dramatically less than the increase that we get when we don't have TH5487, okay? So what does that mean for us? Let's take a look at these answer choices, keeping these 2 points in mind, right? So A, this is just the wild type. This is a standard, the control, or not wild type, excuse me. This is the control that we want to compare to. First point, when we add TNF alpha, remember, when we add TNF alpha, we want to see an increase in NF-kappa B occupancy. Is that what we get? Yes. But why is this one wrong? Because when we add TH5487 on top of the TNF alpha, what we should see is a decrease in NF kappa B occupancy, but instead we don't see any change. The A has to be incorrect. As for B, do we get that increase when we compare to the control? Yes, we get that increase in NF kappa B occupancy, but what's wrong here? When we add the TH5487, we should be seeing a decrease, not an increase, in NF kappa B occupancy, making that one incorrect. Let's take a look at C. When we add TNF alpha, do we get that increase in NF kappa B occupancy compared to control? Yes, we do. Now, what happens when we add TH5487? We do get a decrease, right? That's what we're looking for. We're seeing that we're looking for the decrease in NF kappa B occupancy compared to when we didn't have TH5487. And notably, we also can compare and see that when we have TNF alpha, even though we have TH5487, we still see that increase in NF kappa B occupancy relative to the control. The only comparison that we also have to keep in mind is that it's less, right? We're getting less when we add TH5487 when we're comparing the two groups that have TNF alpha added. So C is looking pretty promising, but as per usual, let's look at the other answer choice here. Immediately we can tell this is probably wrong, right? We have TNF alpha added, but we don't see that increase in NF kappa B occupancy compared to control. And on top of that, when we add the TH5487 on top of the TNF alpha, we should be seeing a decrease relative to this group, but instead we see an increase relative to this group of the NF kappa B occupancy, which makes D an unlikely answer choice and C the correct answer choice for this problem.